So hi, I'm Hanna, and I work at Clinical Genomics in Gothenburg, and I will talk a bit about a research project that I was a part of regarding analysis of circulating tumor DNA in prostate cancer. So yeah, a researcher came to us with her data, and she wanted some bioinformatic help to extract certain information and help to do some visualizations. But I'm going to start to tell you a bit about the background to the project, the biologic background. So liquid biopsy is a non-invasive method to monitor cancer as opposed to tissue biopsy, but it requires sequencing techniques that can detect the very small fraction of circulating tumor DNA or cell-free tumor DNA that can be found in blood plasma. Cell-free tumor or cell-free DNA can be found in blood when cells die. Cell-free DNA is released into the bloodstream, and when tumor cells die, cell-free tumor DNA is released from cancer cells in the same way.
I think we're just looking at the first slide of your presentation.
You can't see the next slide?
No. You're sharing the, yeah, there we can see that.
Okay, I'll keep the presentation like this then.
Okay, good.
You see the second slide now? Or?
Yes, now we see the background. Yeah.
Okay. Yeah, by using continuous blood sampling, you can monitor cancer, and that's a promising application of liquid biopsy. But as I said, one of the challenges is that the fraction of circulating tumor DNA is very small in the blood. So most of the DNA you will find is naturally occurring cell-free DNA. Yeah, it requires very deep sequencing techniques. And the samples that she had in her research project was blood samples from 36 patients with metastatic prostate cancer and 2 healthy volunteers. So cell-free DNA had been extracted and sequenced at different time points. And she also had white blood cell samples from each patient that was used as control samples because white blood cells have no tumor DNA. She had used QIAseq targeted DNA panels by QIAGEN with a panel that was custom made for prostate cancer for sequencing. And she used targeted sequencing of 80 genes split into 847 target regions with known variants related to prostate cancer. And she sequenced according to this QIAseq targeted panel. And DNA molecules were barcoded by UMIs, unique molecular identifiers, before the amplification steps of PCR to make it possible to differentiate between PCR duplicates and true variants. So targeted DNA molecules were tagged by UMIs. So all reads are originating from the same molecule have the same barcode, and then you can differentiate between PCR duplicates and true variants. And UMIs is good to use in this type of case because we have variants in such a low frequency that we want to find. So we want to make sure that they are correct and not sequencing errors. So what she wanted some of our help with in this project was to do some CNV calling, variant calling, and she wanted help with sample checks so that a sample that was annotated as belonging to a certain patient actually belonged to that patient. And we also talked a bit about circulating tumor DNA fraction estimation. But I'm going to focus on the CNV calling part because that's the main part that I've been working on. So in prostate cancer, it's associated with having rather high rates of CNVs. And one common CNV is a gain in the androgen receptor gene, because when you treat prostate cancer, you can use drugs targeting the androgen receptor. And it's common that resistance to these drugs happen after a while, and then copy number gain in the androgen receptor cells is associated with the progression of the disease. So I've used two different methods to do CMV calling. And one is the CLC Genomics Workbench by QIAGEN, which is a tool by QIAGEN for handling their sequencing data. So this was a tool that already existed by QIAGEN, and they had a workflow for comparing white blood cell samples to the patient samples. And yeah, this workflow takes the sequencing reads and maps to the reference genome and removes UMIs and so on, and does CMV detection by comparing coverage from a sample to its white blood cell sample of the same patient. But I have also developed my own CMV detection pipeline, mainly using R. But first, for pre-processing the FOSQ files, I have used the UMI error correct pipeline, which is a pipeline for data analysis of targeted amplicon sequencing data with UMIs, which is what we have in this project. And this pipeline was developed by my colleague Tobias Österlund. And here, in this pipeline, reads that have the same UMI and map to the same gene on the reference genome are grouped together. And then we used a cutoff on group size 2, which means that a consensus sequence is made when 2 or more reads have identical UMIs. So from the outputs from the UMI correct pipeline, I used the information about coverage per base, which I used further when I developed my own pipeline in R. And in this pipeline, the first thing I do is a quality control, because the read counts can vary because of the DNA quality and library prep and the sequencing process. And low coverage is related to poor quality. So the first step is I do a quality control step where I remove samples if the overall coverage is too low, and also remove regions of the sample that have low coverage. And the next step in this pipeline is the normalization step, because for a certain position in the genome, the number of reads that are aligned to that position for a specific sample is proportional to the copy number at that position. But only comparing read depth is not, between samples is not enough to identify CMVs, because sequencing coverage can vary between different experiments. And a number of different factors can have an impact on library preparation and sequencing, which affects read depth. So that's why normalization is important to transform the data by reducing technical biases in the data to make accurate comparisons between different experiments. And for normalization, I have used the R bioconductor package DUSeq2, which is usually used for RNA-seq experiments, and it normalizes reads based on scaling factors. And DUSeq2 normalization uses the assumption that the majority of genes are not differentially expressed, but the purpose of scaling factors and the idea behind the calculations of DUSeq2 are applicable for CMV detection as well, with the assumption that most genes or longer stretches of bases are not amplified or deleted. So normalized counts for each base were computed by dividing the original reads by the scaling factors. And then I do the actual CMV calling in the pipeline, where each sample is compared to the white blood cell sample from the same patient to discover possible CMVs, since DNA from white blood cell sample is assumed to be tumor-free DNA. So for each position, the coverage of the sample is divided by the coverage of the white blood cell sample. And I use the log2 value as a measure of amplification. So if the log2 value is 0, then it means it has the same coverage, and then it's no CMV. And if it's has a log to value of 1, then it's duplicated. And if it's -1, then it's half compared to the white blood cell sample. So that's a measurement that you can use. And you can set a threshold on the log2 value of how small you allow an amplification or deletion to be classified as a CMV. And the good thing about the pipeline that I developed was also that I was able to make some nice visualizations of the CMVs. So I'm going to show you some of the results from different samples. So this plot that you can see here, it has the gene, the androgen receptor gene, and all samples from a patient taken at different time points. And on the x-axis, you have the genomic positions, and on the y-axis, you have the log2 value. So as you can see, for most samples from this patient, the log2 value is around 0. So this indicates that you don't have any CMVs in these samples for this gene. But in 2 samples, you can see that the log2 value is around 3.5, 4, something like that. And this is also the 2 latest samples in the time series taken for this patient. So this is kind of what's to be expected, that it has a gain in the androgen receptor gene. So that's one way that you can visualize the CMVs. And I wanted to compare my pipeline that I developed to the CLC workflow pipeline to see if they both indicate CMVs for the same for the same genes or the same regions. So here I have some different target regions for 2 different genes, the androgen receptor gene and this one. And you can see for these target regions, the fold change is quite high for both the CLC workflow and the pipeline R for the same regions. And if it's around 1, like it is here for this gene, that means that it has no amplification or deletion, because it's the same coverage as is in the white blood cell sample. So even though the values are not the exact same for the copy number, they both indicate that for these regions in the androgen receptor, it's a copy number gain in both methods. And here's another plot that I had to visualize the CMVs. This is you can see the chromosomes on the x-axis, and so each dot in this plot is a log2 mean value for a target region. And if the mean log2 value for the whole gene is greater than 1 or less than -1, the name of the gene is displayed in the plot. So you can see that for this patient, for some of the samples, they have some genes that are just below -1 in the log2 ratio. So this could indicate a loss in these genes. But also you can see quite clearly here, for 2 of the samples, they have quite a high gain in the androgen receptor gene, around 2 and around 4 almost, the log2 value is. So that's another way to kind of visualize the CMV calling results for the samples. So here you can have you can see all genes in the same plot, whereas in the other plot you can do 1 plot for each gene. So that's some different ways to display the results. So that was some of the things that I worked on in this research project. And we used GitHub to deliver our results to the researcher for reproducibility. And so when she's writing her method, she can check what we've done. And if we need to go back and redo something in the future, we have everything described there, our code and our results. So that's kind of a way that we work with research project here and deliver our results to the researchers. So yeah, that's what I had to say.